Creando perfección en la industria de la piratería. Producciones Shark. En MP3. Cumbia.
Prophase is the first step on mitosis. During this step, complex DNA and proteins contained in the nucleus, known as chromatid, condenses to become thousands of times more compact than they were during the interphase. These structures now appears as an egg-shaped bodies, known as sister chromatids. The sister chromatids are pairs of identical copies of DNA joined at the centromere. The centromere is the yellow box at the top, this one. Then the spindle begins to form. This is the spindle, the small yellow things. This is made of long proteins called microtubules. These things are responsible for separating the sister chromatids into two cells.
Cumbia.
Metaphase is a stage in the cell cycle, where all the genetic material is condensing into chromosomes. These chromosomes then become visible. During this stage, the nucleus disappears and the chromosomes appear in the cytoplasm of the cell. During this stage in human cells, the chromosomes then become visible under the microscope. As metaphase continues, the cells partition into the two daughter cells. Cells in metaphase are used in medical research to measure whether all of the chromosomes are present and whether or not they are all intact. This process of looking at chromosomes under the microscope is called karyotyping.
Cumbia.
Anaphase is the fourth phase of mitosis, the process that separates the duplicated genetic material carried in the nucleus of a parent cell into two identical daughter cells. Before anaphase begins, the replicated chromosomes, called sister chromatids, are aligned along the equator of the cell on the equatorial plane. The sister chromatids are pairs of identical copies of DNA joined at a point called the centromere. During anaphase, each pair of chromosomes is separated into two identical and independent chromosomes. The chromosomes are separated by a structure called the mitotic spindle. The mitotic spindle is made of many long proteins called microtubules, which are attached to a chromosome at one end and to the pole of a cell at the other end. The sister chromatids are separated simultaneously at their centromeres. The separated chromosomes are then pulled by the spindle to opposite sides of the cell. Anaphase ensures that each daughter cell receives an identical set of chromosomes.
Cumbia.
Now, we're arriving at the fifth stage of the mitosis, the telophase. Well, and what happens in the telophase? Well, first of all, chromosomes reach the cell's opposite poles. But why do they arrive at the opposite poles? Well, they arrive because they want to decondense, and they begin to decondense. Now, a homologous chromosome arrives to a different pole as well, as you may see. The nuclear envelope material surrounds each set of chromosomes, and here represented in the orange figures. Now, the last step is that the mitotic spindle breaks down, and well, this is an approach of the telophase.
Cumbia.
Cytokinesis is a physical process of cell division, which divides the cytoplasm of a parental cell into daughter cells. It occurs currently with two types of nuclear division, called mitosis and meiosis, which occur in animal cells. Mitosis and each of the two meiotic divisions result in two separate nuclei contained with a single cell. Cytokinesis performs an essential process to separate the cell in half and ensure that one nucleus ends up in each daughter cell. Cytokinesis starts during the nuclear division phase called anaphase and continues through telophase. A ring of protein filaments called the conjugated ring forms around the equator of the cell just beneath the plasma membrane. The contract ring shrinks at the equator of the cell, pinching the plasma membrane inward and forming what is called a cleavage furrow. Eventually, the contractile ring shrinks to the point of there are two separate cells, each bound by its own plasma membrane.
Cumbia. Perfección en la industria de la piratería. Producciones Shark. En MP3.